And so this is, all of immunology and 64 slides. So it's necessarily pretty superficial and fast. And I try to make it not super overwhelming, but immunology is notoriously jargon filled and complex. It's maybe gotten a bit easier over the last couple of years because there's been lots of discussion of immunological principles in the popular press, but happy to go into more detail on any specific topic, either in this lecture or in subsequent lectures. So please, again, don't hesitate to ask more questions about the things that you're working on in particular, or that you're just interested in. And so thinking about what the immune system has to do from the perspective of the immune system's role in fighting pathogens, it has a very diverse target space. It has to deal with intracellular pathogens such as viruses, which we're all now very familiar with, all the way up to very large extracellular pathogens like parasites. This is actually schistosoma that was mentioned earlier in the introductions, which are many, hundreds of thousands to millions of cells collectively, and are going to be much bigger than any individual immune cell. And so it has to develop effector mechanisms and detection mechanisms to distinguish between these different pathogens and develop useful countermeasures to each of them. And part of that involves lots of different pathogen life cycles. So as I mentioned, the intracellular life cycle of a virus, which may be very fast and on the order of hours to days, where it can have exponential replication in a small number of cells, where in a very specific organ, versus certain parasites that have or bacteria that can find specific cell types and stay for days to months to years. To the incredibly complex life cycles of, again, parasitic helminths like schistosoma, where the normal life cycle, even just within the human, is on the order of weeks to mature into the adult worms that are going to cause most of the disease in the liver. But there are phases in the lung, in the skin, in the blood, and all of these are going to have an immunological component. And so one useful way, I think, of thinking about how the immune system functions is by the questions it's trying to answer. So the first question is, when should I respond as the immune system? And so is this protein that I've encountered a self or non-self? And if it's not self, is it actually a threat? Is it something that I have to deal with? And then even if that question is answered appropriately, then it moves to the more complex question of how should I respond, which is what's the most effective means of control, and how do I limit damage to my normal tissue, my normal organs. And so some of the themes that we'll talk about in this first lecture and throughout the course are that the immune system generally takes this path, and whatever immune response we're thinking about, whether it's against a virus or bacteria or a parasite, a very generalized inflammation while information is being gathered, followed by specific pathogen-directed response. And the specificity of the response only grows with time as the response develops. A lot of how the immune system functions is communication via soluble mediators, and these are proteins that are secreted from cell to cell to direct the function of certain cells, the trafficking of certain cells, and the behavior of even non-immune cells. And these soluble mediators, the largest groups of them are cytokines and chemokines, and this is part of the jargon that everyone hates about immunology. These are the interleukins, IL-1 through, I think we're up to IL-40. And chemokines, the CCLs, CXCLs. So just lots of numbers and letters together that you have to memorize and learn how they function in their specific contexts. In every action, there is a feedback regulation to limit hyperinflammatory responses. So there's always a negative loop somewhere in the system with whatever pathway that you're looking at. And there's multiple modes of recognition and specificity determination. So there's enormous amounts of redundancy, and it's not even, it's important to try to avoid thinking of it as designed redundancy. It's not maybe the redundancy you would put into a computer protection system. These are evolutionary redundancies, that convergent evolution that's evolved over time to come up with similar solutions from diverse organ systems. And related to that, it is not centrally organized. So there's not one core decision-making function of the immune system. It's multiple parallel systems acting simultaneously to produce the desired result, cross-talking and regulating each other, but ultimately often arising from very different functional and evolutionary histories. And as a result of that, it's not always clear what's essential, what's redundant, and what is irrelevant. And when you read the literature, you're going to find a lot of papers that, because they're trying to get published in a high impact journal, where they say this pathway is the most important pathway, because if you don't have this pathway and you get this pathogen, you're going to die from it. And so everyone has a figure where their mouse model dies, or their humans are stratified based on disease severity. And what is clear from a number of different studies is that if you really objectively assess which pathways are absolutely essential, it's usually a very small number of what is turned on after a particular infection. But again, the immune system has no way of knowing necessarily upfront what it's being faced with, and so it's going to respond with multiple different effector mechanisms to try to control what is initially an unknown threat. As I've mentioned, with each individual type of pathogen, to be effective, the immune system needs a pathogen-specific approach. So some pathogens have to be eliminated for the host to survive. And this is true generally of small, usually rapidly growing with limited genome pathogens. So these are not particularly sophisticated pathogens in one sense. They're just trying to keep propagating throughout a population as quickly as possible. And they have no chronic phase to sort of cloak themselves and protect themselves and outlast different immune outcomes. And so the goal here is to win a race, to grow quickly enough to transmit before the host dies or there's clearance. These pathogens usually have no idea what's happening in the host. There's no sort of sensing apparatus or feedback apparatus. It's just a maximization of replication. And you can think of this in terms of viruses like influenza, Ebola, RSV, and of course SARS-CoV-2 as well. The timescales here are on order of days. If the immune response fails, it usually fails within the window of days. Some pathogens can vary their acuteness or chronicness depending on the host situation. And so usually these genomes are larger, they're more complicated, and host genetic and environmental factors might determine susceptibility and permissiveness for these different outcomes. And there's bacteria like this, like TB, that can have what you would think of as maybe like a classic bacterial life cycle where it's just replicating in cells and trying to get out, and almost like a virus just spread to the next person, but then can also go long-term chronic and knows something about the metabolism of the host and the current stress level of the host to know whether or not it needs to be in one of those two phases, and also even relatively simple viruses like HCV. And then some pathogens are almost exclusively chronic, and again, not always, but usually these are much more sophisticated genomes, very large size genomes that have a lot more interaction with the host and feedback from the host. What determines chronicity here is not always fully understood in every circumstance. And it could be that the host has actually just not evolved a mechanism for effective clearance. Pathogens, because relatively they're all going to have a smaller genome than the host, can evolve a bit more quickly. And so maybe just ahead of us in the current evolutionary arms race. These are usually not acutely lethal. So you live a long time with certain parasitic worms, with even with TB, and you can even think of HIV and somewhat in this category, though it doesn't have a very large genome. So putting that into this little chart, we're thinking about the stages of an immune response and again, the generality to specificity trend and the goal of trying to block infection at as early a stage as possible. So ideally, most of your infectious encounters are actually blocked upfront without any involvement of what we would call the professional immune response by the bulk removal of the infectious agent. And then if that fails, the initial response is going to be driven by inflammation, recruitment, and activation of effector cells that are somewhat nonspecific, and again are with the goal of just removing the infectious agent before it can move into an exponential replication cycle, and before there's significant damage to the host, and before there's actually significant energy expenditure in a highly specific immune response. And then failing that, there's what we normally think of when we talk about immunity, which is the recognition by the adaptive immune system, clonal expansion and differentiation of those cells, which is an incredibly energy-intensive process. These are also things that make you feel very, very sick, but then are generally more effective and targeted at removing an infectious agent, and can remove an infectious agent that is now in an exponential phase of growth. So these stages, to put them more into specific words, start with the physical prevention of infection. And this is regulated by things like your skin, mucus in the lungs, and then the mucosal surfaces, surfactants, and cilial beading. So you're constantly inhaling lots of different types of bacteria and viruses, and they are getting stuck in the mucus in your lungs. They're getting quickly removed by the coordinated beating action of the cilia on all of your lung epithelial cells. They're going to push those back up, and you cough a little bit, and you actually avoided a very productive infection. When that fails, you get potentially some epithelial inflammation. Epithelial cells are the cells that form your tissues. And so in your lung, when you think about your big pink heavy lungs, most of those cells are epithelial cells that are forming the tissue, and they're what's actually infected with influenza or SARS-CoV-2, and they will try to fight off the infection initially, and they can actually exert some level of control. They can also fight off some bacterial infections as well by secreting proteins called defensins. They will also start that early inflammatory response by secreting some cytokines, including interferons, which are going to be these soluble mediators, again, that are going to start letting neighboring cells know that there's a problem, and also alerting the professional immune system that they might need to get involved. So after these fail, the pathogen's still replicating, you call in the professional and innate cells, and these are going their purpose is to deal with foreign pathogens. And so they're going to do things like eat the foreign pathogens. So they're just going to be non-specifically eating in areas of inflammation. They'll start amplifying some of these signals coming from the epithelial cells, and they can actively kill the pathogen as well, but usually with some element of tissue damage involved. And then finally, if all of this fails, you'll have the generation of the adaptive response and clearance by antibodies, CD8 T cells, and CD4 T cells, either via extracellular or intracellular killing of the pathogen. And we'll talk about specific examples of all of these as we go. So we often think of the immune system in terms of the cells that are involved, and we have this divide between innate cells, which are non-specifically targeting broad classes of pathogens, and they're listed here, including phagocytic cells like macrophages, dendritic cells, and so forth. Granulocytes such as eosinophils and neutrophils. And then on the right side of the graph here, we have the adaptive immune cells, so B cells and T cells. And T cells will talk mainly about CD4 and CD8 T cells. There are these cells that sort of bridge between these two, and in the course of these lectures, we're not going to cover them very much, but if you have a particular interest in either any of these populations that we're not covering, we can try to cover them. Okay, I've had a request to slow down a bit. I will do my best, but it's a pretty packed lecture, and so we are recording it, and so people can watch later. I just want to make sure we hit on a number of topics that are going to be important for the modeling that come later. So this is just a quick slide on how the skin and mucosal surfaces can serve as an initial blockage of pathogens coming into the system. So your mucosal surfaces are generally your skin, your gut, and your lungs. You can think of these as the surfaces where you're really exposed to the outside world. The skin is obvious, the lung is where you're breathing, the gut is where you're eating. And all of these are made up by these epithelial cells, again, that are tightly packed in with each other and are trying not to let anything through into your bloodstream. They secrete all kinds of both chemicals and protein products that are there to kill bacteria and control the bacteria that do grow there. And we've had a couple of people mention they're working in the microbiome space, and all of these surfaces actively regulate the microbiome that they are colonized with. And we've studied that mostly in the gut, but a lot of work coming out now on the skin and the lung. As I've mentioned, the a lot of immunology is cellular immunology, meaning we study the behavior, the trafficking, the expansion, and the activation state of the different cells. And all of these cells are that we study are generally derived from hematopoietic stem cell progenitors. So you have a cell in your bone marrow that can give rise to all of your adaptive immune cells, including your B cells and T cells, and then all of your other innate immune cells in the blood, including actually your red blood cells, and then your macrophages and dendritic cells and granulocytes. And so all of this is part of the same system, all from the same progenitor, and all can be damaged by any kind of insult to the bone marrow. Okay, so now we're going to move through the stages of an immune response, sort of in the order in which the recognition occurs and the effector mechanisms are mounted against a particular pathogen. And so the very beginning of the immune response after a pathogen has gotten through these barriers at the mucosal surfaces involves innate immune pattern recognition. And this conceptually actually is a really nice feature of immunology history, and that these pathways were really recognized and developed theoretically from a true hypothesis-driven approach. The person who's mostly credited with kind of founding the field of innate immunity in the modern era is a man named Charlie Janeway, who died a number of years ago. But he wrote a very important article on what he called pattern recognition, which was obviously a concept in information theory at the time. But he had the idea that there had to be proteins in the immune system that could detect generalized non-self patterns and mount and trigger inflammatory responses. And his lab, in collaboration with Ruslan Metsadov, who was the senior scientist in his group at the time, went on to define these pathways, some of these pathways. And Ruslan Metsadov, along with several other people, went on to win the Nobel Prize for this. Ruslan Metsadov did not win the Nobel Prize for this work, which was quite controversial. But and Charlie Janeway had passed, but there was a Nobel Prize awarded for innate immunity about 10 years ago now. And the idea here is that there are these proteins in your immune system, and there's three big classes that we're going to talk about here, which are the TLRs, which stands for toll-like receptors, the NLRs, which stands for nod-like receptors, and the RLRs, which stands for Regi-like receptors. And their purpose is to recognize patterns, molecular patterns that are present in foreign pathogens, but not in your normal cells. And so things that they might recognize are things like lipoproteins, so proteins that are modified by specific fatty acid structures that you don't make normally in your body, but that bacteria make all the time and that are critical components of the bacterial cell wall. There are also even recognition of specific proteins that, again, you don't make in your body, like flagellin, which is the tail on certain bacteria, but that bacteria make and have to survive. And so you've evolved these recognition molecules that recognize broad classes of pathogens, bacteria, viruses, parasites, and initiate in non-professional immune cells and in professional immune cells an inflammatory response mediated by signaling pathways like NF-kappa B, if you're familiar with that, and cell biology, and the type 1 interferon response pathway. And so some of these proteins are on cell surfaces. And again, these can be on normal cells, they can be on professional immune cells, and they're just sort of always looking and always surveilling for specific patterns of these pathogens. And then some of them are inside the cell, and they're looking for intermediate stages of pathogen life cycles. So in terms of viruses, there are proteins that recognize viral genome intermediates, so things like double-stranded RNA, which is something that you shouldn't make a whole lot of in the normal life cycle of any of your cells, but is a critical intermediate in a lot of viral replication cycles. Even certain forms of single-stranded RNA or DNA in the wrong part of the cell can be a pattern that's recognized by these pathways because they are patterns that are out of place in where you keep these things in your cell. So if you have DNA in an endosomal vacuole out in the cytoplasm of your cell, that's not where you keep your DNA, right? Like you keep your DNA in the nucleus. And so that probably means you have a DNA virus somewhere in your cell. So this figure is a nice figure. I like it still because it shows actually that a lot of these proteins converge on very similar pathways in terms of activating the cell and inflammatory processes in multiple cells. But these pathways have just kept getting more and more complex. There are 10 to 11 TLRs, depending on the species that you're looking at. There are wildly varying numbers of NLRs, around 30 in humans, and these are all cytoplasmic. And then there are expanding numbers of members of the RLR family, which looks at nucleic acids, and many different complex signal transduction pathways that they all activate. But if you look at this, you'll see there's a lot of commonalities here. So again, NF-kappa B is activated by multiple pathways. The IRFs, the interferon response, are activated by multiple pathways. And for viruses in particular, and we'll be talking about flu and RSV and SARS-CoV-2, the recognition of nucleic acids is really critical. And so double-stranded DNA and the cytoplasm is a really key feature that will activate a number of different recognition molecules. So again, there's this huge redundancy where you're recognizing these patterns in a relatively non-specific way by multiple pathways, because your cells are constantly being bombarded by pathogens that are trying to steal resources from your cells and use very similar strategies in terms of double-stranded RNA and double-stranded DNA intermediates and their replication cycles. So again, lots of details we can go into on any of those pathways if people are interested, but at the high level, what happens after this recognition occurs, you get signaling through the cell, propagating down to transcription factor activation that's going to turn on a transcriptional program in the epithelial cells of the tissue that you're in and in the professional innate immune cells that have come to deal with the inflammation that's occurring. And for viruses in particular, the classic response and one of the very earliest cytokine systems that was discovered was the interferon response pathway. And the type 1 interferons that are the classic signature of an antiviral response are interferon alpha and interferon beta. So these are the names of proteins that get secreted from a cell that's infected with a virus, that's recognized that it's infected with the virus via these pattern recognition receptors. And what these do is they get secreted from the cell, and then they can alert other cells nearby that have receptors for interferons on them that there's a virus in the neighborhood and that it's replicating. And these cells can start turning on other pathways that will start inducing resistance to viral replication in all cells. This is a typo from the textbook. It can increase expression of certain proteins that will make the cells more visible to the adaptive immune system and activate other elements of the innate immune response. And so this is just within hours of a virus entering your body, you'll start having higher detectable levels of type 1 interferons. As part of that now, you've got inflammation in your tissue, you have cells, professional innate immune cells starting to come in, and they're going to start trying to deal with this infection. But again, it's really a very broad class of infection now. We know we have a bacteria or we know we have a virus, and we're going to try to deal with that in a sort of class-specific manner. And so one effector mechanism that's commonly used by the innate immune response is digestion and chemical destruction. So we're just going to try to eat everything in the extracellular space that is suspect, and we're just going to try to destroy it. But this is going to cause even more inflammation and potentially tissue damage. And so this is potentially problematic. And so you have cells like macrophages that will go into say your infected lung, and will literally just start eating things in the environment and can put those things inside endosomes, little vacuoles inside the cells, where it will release reactive oxygen and nitrogen intermediates that will destroy anything in that vacuole by chemical digestion. As part of this process, it's going to trigger more of those innate immune receptors that are pattern recognition receptors that will broadly determine that there's an infection and drive the secretion of even more cytokines, amplifying the signal of inflammation. And so these are the early phase cytokines that are released that drive non-specific inflammatory responses. So IL-1 beta, TNF alpha, IL-6, CXCL8, and IL-12. These are cytokines produced by both stromal cells and professional innate immune cells that are going to recruit even more innate immune cells and also cause local and systemic effects that will be helpful to the immune response in trying to target a pathogen. And so these include things like raising your temperature, which can interfere with pathogen replication. And so that's what causes your fever. It can also make your vascular endothelium a lot more leaky. And the point of that is to get more cells into the tissue to deal with the infection. And so this is a little bit of what that looks like. Imagine this is a vein or an artery or a blood vessel. So you've got cells cruising along here. They're going by your lung, let's say, and this is the border of that blood vessel. And if there's a local inflammation in the lung nearby that blood vessel, all of these chemokines and cytokines are coming through that wall and telling professional immune cells that are going nearby that they need to come and help out and deal with these pathogens, which are these little, I guess these little yellow things here. And so what will happen is the cells will adhere to the blood cell wall and they'll cross through into the tissue where they'll start doing their function. And this is what causes the redness and swelling around an area where you have inflammation, because you have liquid and cells leaking out of your blood vessels to come into the tissue to get rid of your pathogen. For the sake of time, I'm not going to spend a lot on this other pathway, but I like to use it as an example of the convergent evolution of the immune system. There's a whole other innate pathway of recognizing pathogens called the complement pathway. There are multiple ways to activate the complement pathway, but the complement pathway is a collection of proteins that are always circulating in your blood at high concentration at all times, and permeating your tissues at high concentration at all times, and they're totally inert most of the time. And so as they are circulating around, there are triggers that can activate the complement pathway. They can be triggered by antibodies, which we're going to talk about actually towards the end of the talk. So adaptive immunity can feed back on innate immunity, or they can be triggered actually directly by structures on pathogen surfaces, sugars and other structures. And again, this is another form of pattern recognition. And what happens is it starts this cascade of, once the first complement protein has been activated, it will start activating the subsequent parts of the cascade. There are about 11 proteins in the complement cascade that each have regulatory checks on them, so that you can think of it as sort of a series of dominoes that once one starts to fall, they all start to fall. And the end result of it is that the complement proteins can actually directly kill the pathogens, and they can actually, they recruit inflammatory cells. They can almost act like cytokines themselves, and they can actually kill infected cells. And so they can actually cause quite a bit of inflammation and tissue damage if they're improperly regulated. And there are complement-mediated autoimmune diseases where complement's kind of gone off the rails. But what this looks like is you've got these loose proteins, complement proteins, indicated by the C here, floating around. When they finally get activated, they start forming these very complex structures, including this structure called the membrane attack complex, which is a pore that will just insert itself into the membrane of a pathogen or of an infected cell, again, only once the complement's been activated in the proper way. But this is what this looks like. If you're looking at a cell where complement's been activated, hundreds of thousands of pores can be pushed into that cell in a matter of seconds. And so it's incredibly destructive. It's like a nuclear bomb onto that cell, where it just is completely ripped apart again, by your own host factors, once that cell has been targeted for destruction because of recognition, a pattern recognition that something is wrong with it. And then one of the final parts of innate immunity that I want to talk about is something that we call natural killing. And this is now starting to bridge between innate and adaptive immunity, from pattern recognition to really specific pathogen-specific targeting. And what natural killing does is mediated by a cell type called a natural killer cell, which is a lymphocyte, which is the same type of cell as our B cells and our T cells that are going to mediate the adaptive immune response. So those are going to be the cells we're going to talk about in the second half of this talk, that are really formed to make very specific responses. And one of the key ways that T cells know which cells to kill is by monitoring a set of proteins on your cell's surfaces called MHC molecules. And we're going to talk in detail about what MHC molecules are, but they're a way for T cells to surveil what's happening in normal tissue. And so viruses have figured this out over the years. Bacteria have figured this out. And herpes viruses in particular have figured this out very well. And so they know that one of the ways that T cells are seeing that they have infected a cell is via these proteins called MHC molecules. And so what they do is they actually make proteins that go in and grab onto MHC molecules and rip them inside the cell so that the T cells can't see that the cell is infected. So that's these yellow molecules here, the MHC molecules. So that's, an escalation on the virus side of things where they say, well, we're going to just make it so that you can't see what's happening. And the T cells are now not going to work. And so what we have to counteract that is the natural killer cell, which goes around just looking at whether or not cells have MHC molecules or not. And if they don't have MHC molecules, then the natural killer cell gets activated and it kills the cell, because it basically says something funny is happening here. The cell's trying to evade being monitored by the adaptive immune system, so we're just going to get rid of it because it's causing trouble. SARS-CoV-2 downregulates MHC class 1 molecules as well, and it's probably one of the reasons why virus can propagate so long in some people. And cancers also drive down the expression of MHC class 1 molecules, again, just to avoid recognition by T cells. And so there's actually a whole network of these innate lymphocytes. NK cells are just one of them. But again, for the sake of time and the talk today, I just give that one example. And so now in the second half of the hour here, I'm going to talk about adaptive immunity, which involves professional T cells and B cells that are going to be hyper-focused on a specific pathogen. And so to get to this adaptive immune stage, it means these two parts of the immune response have at some level failed. They might have held off the replication of the pathogen for a period of time so that you didn't get very sick and you didn't die and you didn't get overwhelmed by the pathogen, but there's still pathogen around and we need our adaptive immune system to actually rid us of it for once and all. Okay, so adaptive immunity, it's going to be mediated by B cells and T cells, part of the hematopoietic system here. And B cells and T cells are the effectors of the adaptive immune response. And there are three broad arms of adaptive immunity. So B cells make antibodies, which are soluble effector molecules. These are molecules that are secreted from cells, will circulate through your tissue and through your blood, and can actively block pathogens and remove pathogens from those sites at high concentration. And we've all heard a lot about antibodies, I think, over the last couple of years. And then T cell immunity is all cellular based. There's nothing, there's not this soluble effector. And so CD4 T cells, I describe as the quarterbacks of the immune system. They're really key to coordinating all of the other host responses, even the innate response, CD8 cells and B cells. Once CD4 T cells are on the scene, they are dictating the quality of the response and the specific effector mechanisms that are being used to get rid of a particular pathogen. And then CD8 T cells are the killers of the immune response, and they can kill an infected cell directly. And so as a result, they're only useful against intracellular pathogens. So viruses, bacteria that live inside the cell To get rid of them, you really need a CD8 T cell to go and kill all of those cells that have been productively infected. But CD8 T cells are not going to be very useful against a very large pathogen like a helmet. All three of these pathways have memory, and that is a key feature of what we think of when we think of immunity, is that once they've seen a pathogen, if they see that pathogen again, they respond more quickly and with a higher magnitude, much more rapid and efficacy. And so this summarizes that sort of these two sides. Humoral immunity is another word for the B cell mediated immunity, where we have a B cell that will secrete antibodies that are highly specific for a given pathogen. As we know with SARS-CoV-2, these antibodies are specific for substrains of that pathogen. So we have Omicron-specific antibodies versus Delta-specific antibodies and so forth. And then we have our T cell mediated responses. So we have our helper T cells, which are our CD4 cells, and our cytotoxic T cells, which are our CD8 cells that are going to kill an infected cell and remove the pathogen reservoir. So B cells and T cells generally live in the lymphatics in your body, which are a parallel system in a sense to your blood system. There are lymphatic vessels that connect large sacs of CD4, CD8, and B cells that we call lymph nodes. And so this is where your resting adaptive immune cells live on their day-to-day basis when they're not doing anything special, when they're not responding to a pathogen. This is where they like to live, and this is where they wait to determine whether or not they're going to have to respond to something. You all, I think, are probably familiar with your lymph nodes, but you have hundreds of lymph nodes throughout your body, but we often talk about the ones, our cervical lymph nodes in our throat that can get swollen, or under your arms after you had a vaccine. And so these are distributed throughout your body, ready to respond, again, mainly to infections from those mucosal surfaces. So they're well positioned to know what's happening at those mucosal surfaces. Okay, so just like I kind of mentioned that there was a theory that undergirded a lot of our discoveries in innate immunity, there was similarly a theory that undergirded our understanding of adaptive immunity, and that was the clonal selection hypothesis, which was proposed by an Australian, Macfarlane Burnett, in the middle of the 20th century. And what he was working from was the observation that we often don't get the same infection twice. And he was trying to figure out why we don't get the same infection twice. And they also, at that time, had some understanding that there were factors in the blood that you could use to transfer from one individual to another individual that would prevent the second individual from getting an infection that the first individual had gotten before. And so he wrote this theory up called the clonal selection hypothesis before we even knew that there were lymphocytes, before we knew that there were T cells and B cells, and came up with this theory of how this adaptive immune system would work that ended up being remarkably accurate. And the key postulates of this hypothesis were that you would have a cell, which we'll call a lymphocyte, that bears a single type of receptor with a unique specificity for the pathogen that we're going to try to deal with. So each cell has a one receptor type that is specific for one pathogen. The interaction between the foreign molecule, the antigen from that pathogen, and the lymphocyte receptor with a particular affinity will activate the lymphocyte. Once that lymphocyte is activated, it will proliferate, and each of its daughter cells will have the same antigen receptor. And this was key. It's a way of amplifying the recognition signal. So I had one lymphocyte with one antigen receptor that could see that antigen, and now I have a million because it's proliferated and every offspring has an identical receptor. And there had to be a way of getting rid of lymphocytes that saw self and that could cause autoimmunity. And basically, the details obviously took a lot to be worked out, and there's still things we don't understand, but there's aspects of every single one of these that are absolutely true, and fundamentally the idea was correct. And so if we think about it from the B cell side of things, the B cell is the lymphocyte. It starts out with a surface receptor, which is specific for a particular antigen. And if it sees that antigen, say on a virus, it will activate that cell and it will start proliferating here. And every daughter cell will have that same receptor, which eventually becomes the secreted antibody that we've been talking about. So this structure will get replicated many, many, many times, and eventually you'll have a clonal population of cells that are responding to a particular antigen, this orange antigen, whereas this green B cell here has nothing to see, and so it just sits in the lymph node happily, not doing anything because it hasn't seen its antigen. So a key thing that people didn't understand until really the 1970s or so was how the system could work, because how do we make all of these distinct antigen receptors that can see all of the vast array of antigens that we'll encounter in our lives, including antigens that are purely synthetic, that have never been seen before in the history of human evolution, and have that all be encoded in their genome in a way that it's heritable, so that as the cells proliferate, they keep the same antigen receptor. And what was discovered in the 70s was that uniquely in B cells and T cells of the immune system, your genome changes as those cells develop. And so this is a very, very old school technique called a Southern blot, where they were looking at the region of the DNA that coded the B cell receptor, the antibody. And they found that if you looked in that region in a normal cell, like in a skin cell, you had this pattern in the DNA. And we can go into what a Southern blot is, but nobody does them anymore. But it's kind of an old low resolution way of doing PCR. And if you looked in a B cell, the DNA was organized differently. So this also won the Nobel Prize. And what it demonstrated, just these four bands here, what they demonstrated was something called somatic recombination, which means in a developing B cell, the DNA that determines the antibody expression by that B cell actually gets chopped up and rearranged. And we're going to talk about that in a bit more detail, but that's the concept of how we generate the diversity of antigen receptors. And so to get into that, I'm going to first just describe what a B cell receptor or an antibody looks like. And it's going to be very similar to what a T cell receptor looks like. And so you have two chains. And so this is this one chain here, these four blocks here. And we're going to call this the heavy chain because it's the larger of the two chains. And then this is the light chain, it's the smaller. So there's these two blocks. So light chain, heavy chain, and then that's duplicated. It's the same proteins on the other side, heavy chain, light chain. So you have two light chains, two heavy chains. The two heavy chains are identical. The two light chains are identical. You have a region of the antibody that's determined by the heavy chain that we call the constant region. It's the same across all the antibodies. That's why it's constant. And then you have the variable region, and this is going to be the business end of the antibody that binds to the antigen. So this red right here is what's going to stick on to your SARS-CoV-2 or your flu or your TB. And this is the hypervariable region of the antibody. So how do we make this? It's that somatic recombination process where we're actually going to chop up the DNA that encodes the antibody receptor, and we're going to stitch it back together in a way that's purposefully error prone to create a lot of diversity so that every B cell has a different antibody and those antibodies all see different things. And so what we start with are these large cassettes of gene segments, and we call them the variable regions, the diversity regions, D, and the joining regions. And so you have, I'm going to skip ahead one slide, you have 40 copies, slightly different from each other, of these different variable regions, 25 diversity regions, 6 joining regions. These numbers actually change between different people, and we discover new ones all the time. And when you're a developing B cell and you're figuring out what antibody you're going to make through a semi-random process, you pick one of these V regions, one of these D regions, and one of these J regions. And you have special enzymes that come in and cut your DNA and then fuse the V region, the D region, and the J region that you picked together. And it cuts out a lot of the other ones that you're not using. And you end up with this fused V, D, and J in your genome. And that's what the Southern blot had shown, was that there was a whole bunch of DNA cutting and stitching back together. When you do this, you actually purposefully make mistakes so that there's lots of mutations here, and you end up with almost a random sequence of receptor here that will be able to bind lots of different antigens. And so you do this in every B cell. Every B cell gets a unique B cell receptor, but it's now encoded in the DNA, and you don't do this cutting thing outside of the development of the cell. And so after it's fixed, now every cell that divides from your starting B cell will have exactly the same receptor, but its receptor will be different than every other B cell that developed at the same time. So this generates an enormous amount of diversity. So you have these different variable regions, D regions, and J regions, and then you purposely make mistakes when you put them together. And so if you add up the sources of diversity for either antibodies, which we also call immunoglobulins, or T cell receptors, which are over here, you get somewhere between 10 to the 13th and 10 to the 18th potential unique T cell receptors or B cell receptors. And these are almost certainly, this is from 2005, massive underestimates. A lot of these numbers are based back on numbers that were found in the 80s. And so the true number of potential diverse receptors is really on the order of probably 10 to the 40 to 10 to the 60, these are gigantic numbers. And you only have about 10 to the 12th of these cells in your body. So we're actually massively undersampling the potential diversity of the system in any individual human. And so 10 to the 12th potential unique receptors drawn out of this much larger pool of potential TCRs and BCRs. So antibodies are even a little bit more complicated than what I've just told you. So you have the constant region, as I mentioned, and the variable region. Annoyingly, the constant region can change, so it's not always constant. So you can use different constant regions, and the B cell will actually switch which constant region it's using based on its current function. And so all of B cells start by using a constant region that we call IgM, and you can see that here, these 3 blocks. And then they'll switch through subsequent constant regions that serve different purposes. And so this will actually come up in some of the modeling because, for instance, IgA is a form of an antibody that's much better in terms of being secreted at mucosal surfaces, like onto your skin and into your lung. The variable region actually stays the same now, and the constant region changes so that the antibodies can function differently in different tissues. So to sort of take you through the life of a B cell, as it's developing, it forms its unique receptor by rearranging its immunoglobulin genes, that VDJ recombination. It then puts that antibody on its cell surface, and it's in this IgM form, and 99.9% of your B cells will live their whole life like that, and nothing else will ever happen to it because they will never see their antigen. The one lucky B cell that happened to be SARS-CoV-2 specific will see its SARS-CoV-2 antigen there. It will bind and activate. It'll start causing that switching of the constant regions that it'll use, and it'll start proliferating and activating the cell. So now you're going to have millions of these B cells that all have this specificity for a particular antigen. And eventually it will differentiate into what we call a plasma cell, where instead of just keeping the antibody in its cell surface, it will secrete them like little torpedoes that will go out into all of your tissues and directly bind the pathogen and interfere with its ability to live its life. So it will coat the pathogen with these antibodies, and now the pathogen proteins won't work, it won't bind your cells, and it will actually trigger those macrophages and other innate immune cells to come eat the pathogen. And so this is the way that antibodies actually prevent you from getting infected, because they'll just coat the pathogen as soon as they enter the body. I'm going to skip over this. I mentioned earlier that there's specific types of antibodies that do better in certain parts of your body. So IgA is an antibody that is designed or has evolved to specifically be secreted into mucosal surfaces. So this is a picture of your gut, actually. And so your plasma cell B cell that's making an IgA antibody will sit underneath your gut in the tissue. will secrete the IgA, and that'll get traversed across the epithelial membrane and accumulate in very high concentrations in your gut. And this is actually one of the key ways that we regulate our microbiome. So you also have these B cells in all of your other mucosal surfaces in your gut and your lung and the genital mucosa. One unique thing that B cells do that T cells don't do is after a B cell sees its antigen, it will actually try to make the antibody better. So it doesn't do the whole rearrangement thing again. It doesn't do the VDJ recombination, but it will make specific point mutations in the antibody gene to try to increase the affinity of the antibody for its antigen. And so over the course of an immune response, you will go up potentially almost 2 logs in the affinity of the antibody for the antigen through this processive cycle of B cells mutating their antibodies, testing whether that antibody can still see the antigen in the context of a lymph node structure called the germinal center, that will, if the B cell gets better at seeing the antigen, give it a survival signal. If it gets worse at seeing the antigen, the B cell dies. And you have millions of B cells now that can see this antigen, and so you only need a few of them to get better. And you, as the antibody gets better, those B cells will continue to expand. And this is really what's happening in a vaccination response, and why vaccination responses take so long to mature, and why your vaccination response keeps getting better even months after the vaccine. It's because this process is happening and making your antibodies better. This is a reference for you for the different types of immunoglobulins, that the different constant regions that I mentioned. I briefly mentioned IgA. I've mentioned how all cells start out as IgM. And we can talk about these in more specific examples as we go into the pathogen-specific talks. Okay, in the last 10 minutes or so here, I'm going to do T-cell mediated recognition. So this is an antibody. We went through how antibodies are made. T-cells are very, very similar structurally, evolutionarily, but they are always cell-bound. They are never secreted. So they're never the effector molecule themselves, because they're always just on the cell surface, and they're used by the T-cell to recognize a foreign antigen. And I've mentioned this already, but the way T-cells recognize foreign antigens, conventional T-cells, which is what we're going to focus on here, is in the context of another protein on the target cell that it's trying to look at, called MHC. And so MHC stands for major histocompatibility complex. In humans, we call it the human leukocyte antigens, or HLA. Those terms are used interchangeably. I know it's very frustrating, but every cell in your body has an MHC molecule on it. And you can picture it like a little box that's sitting on the cell surface. And there's you have thousands of these on your cell surface. And what they're doing is taking little pieces of proteins from inside the cell and putting them on the cell surface to say, this is what's happening inside my cell right now. And that makes it very easy for the T cells to come along and surveil what's happening inside the cell. So the T cells know to just look inside this box, the MHC, on the cell surface. This is kind of looking at it from the side. This is looking at it from the top down. And the T cell receptor is going to look just directly into that box and say, what's happening in the cell? Do I see my antigen? Do I see a piece of a virus, or do I see a totally normal self molecule? And just like B cells, every T cell has a unique T cell receptor as a first approximation, and T cells are going to only see one particular antigen, and if they see that antigen, they'll get activated and perform their T cell functions. It's just some more pictures of the MHC here. And there are 2 big types of MHC molecules. There's MHC class 1, that's on every cell in your body. And there's MHC class 2, which is only on professional innate immune cells. MHC class 1 binds to CD8, and that's what makes a CD8 cell see every cell in your body and be able to kill every cell in your body that's infected with the virus. Whereas MHC class 2 binds CD4, so you only are going to have professional innate immune cells talking to CD4 T cells, and those CD4 T cells were the quarterbacks of the immune system. So remember, they're sort of the master regulators of everything that's going on, and they really are only going to see antigens from other professional immune cells that are highly regulated and very careful about what types of immune response they're starting. Whereas the CD8 T cells are going to be able to go out and kill every cell in your body if it's infected, which is what you need when you're infected, say, in your lung with a virus. So I mentioned you've got this random process generating these antigen receptors, and there's no sort of prime mover telling this T cell you're SARS specific, you're flu specific, and so forth. It's a random process. So you're going to generate receptors that are actually specific for self. So how do you avoid having massive autoimmunity from all of these T cells that were randomly generated to see a self protein? And so you actually have a structure in your body called the thymus, which is where T cells develop, which is why they're called T cells. It stands for the T in thymus. And the thymus is a proving ground that educates the T cells and removes the self-reactive cells from your body. So as the T cell's developing, it randomly makes a receptor. It may be specific for something you'll never see. It may be specific for a virus. It may be specific for a self-protein. If it's specific for a self-protein, it will see that self-protein in the thymus because the thymus is really good at expressing lots and lots and lots of different self-proteins. And T cells wash over all of those cells and look and say, do I see anything I recognize here? And if it does, that T cell kills itself during development. So it never gets out of the thymus. If it looks through everything in the thymus and doesn't recognize anything, then it can leave the thymus and go and live in a lymph node and hope that it's specific for a virus that you'll come across at some point later in your life. To start an immune response, we can't just start an immune response with any cell. And again, this is you're talking now in adaptive immunity about cells that can cause massive amounts of tissue damage. And when adaptive immunity goes wrong is when you get really profound autoimmune diseases like diabetes, rheumatoid arthritis that can be lethal. And so to restrict that from happening as much as possible, adaptive immune responses on the T cell side can only be initiated by presentation, MHC presentation from professional antigen presenting cells. So these innate immune cells that are tasked with being the cells that determine that there's enough of a problem, enough inflammation, enough of this pathogen to get T cells involved. And the most potent antigen presenting cells are dendritic cells. So these live in your skin, in your lung, and they're loaded with those toll-like receptors and other innate immune pattern recognition receptors. And they're going to make a decision that there's enough inflammation and enough of a pathogen to go to a lymph node and activate a naive T cell and tell that T cell, we need help. And so it will search out the cells that are specific for those antigens and license that T cell to now go to the tissue and actually kill target cells without having a professional innate immune cell around anywhere. And so this is what we call the T cell synapse, where you've got some piece of a virus or a bacteria presented in the context of MHC that's visualized by the T cell receptor and determined, this interaction is determined by this co-receptor, either CD4 or CD8, and that activates the T cell and it will proliferate, and every daughter cell will have the same T cell receptor, and again, we'll have an army of very specific cells. I know we're at time, but I only have a few slides left, and I'll skip to the end shortly. So I've mentioned there's these professional innate immune cells that can get the immune response started, dendritic cells, macrophages, and then potentially oddly to you, you might be surprised to hear that another professional cell that can activate a T cell is a B cell. And B cells can talk to T cells and present antigen to T cells in a professional manner. But again, it's another way of restricting that activity to professional immune cells. All of this happens in the lymph node where you have B cells and T cells living in different areas, but they come together in the context of inflammation to talk to each other to organize the immune response. The last concept that I want to convey to you is T cells have a broad range of effector mechanisms that they can employ depending on what type of pathogen we're dealing with. When we're dealing with intracellular pathogens like viruses and certain bacteria, the response that we want is something called a Th1 response. So that stands for T helper 1 or a type 1 response. And that's going to be a type of response that is highly cytolytic, meaning we're going to go out and try to kill all the infected cells. And it's because it's an intracellular pathogen. It's a virus that's replicating inside a cell. It's a bacteria that's replicating inside a cell. And we're going to try to get rid of all of those cells. And so we're going to have CD4 T cells that organize as quarterbacks of the immune system, this type 1 response. And CD8 T cells are going to be really critical here because they can go out and kill all those infected cells that are expressing MHC class 1. If we don't have an intracellular pathogen and we have a big worm, for instance, CD8 T cells are not going to be helpful because the worms don't have MHC class 1 on them, and they're not going to be able to be targeted. But antibodies can be helpful. And so we need lots and lots and lots and lots and lots of antibodies. And so we're going to have a different type of response that we call a Th2 response, where CD4 T cells are going to talk to B cells primarily and say, you need to make as many antibodies as possible, a certain type of antibody. It's very good at getting rid of worms. And that will drive that response. Now, this is a very simplified worldview. There's not just two types of immune response anymore. About 10 years ago, we added a third one that we call Th17 responses. You might say, why didn't we go to Th3? This was actually to make it a little bit easier because the signature cytokine, soluble mediator that's signaling the type of immune response that's involved was IL-17. This is for certain types of bacterial responses, including strep pneumoniae, which we heard about a little bit in the introduction. And now there are actually many more types, but for the sake of our discussions here, we'll stick mainly with these three. And as I mentioned, just like B cells, once we've activated these cells, they're licensed, they're going to go out and start performing their effector function. And so if I'm a CD8 T cell engaged in a type 1 response, I get primed by my dendritic cell in the lymph node, and then I'm going to travel to my epithelial surface, say in the lung. I'm going to see my infected cell, and I'm going to kill it, and I'm just going to keep moving down the line and killing as many cells as I can. This just summarizes a lot of what I've just told you. A lot of how we characterize things in immunology and what you'll have as data when you're modeling, particularly from humans, is either counts of these cells or measurements of the cytokines and the molecules that they make in the blood. And obviously we can't take apart humans and find out exactly what's happening in their immune response. So we read these out as a reflection of what's happening immunologically. And then these last slides here are really just summaries to help you as references, and we can refer back to them during some of the specific talks, but points out that there are these different effector molecules of humoral immunity and cell-mediated immunity that work against different viruses, bacteria, these different life cycles of the immune response. And for the sake of time, I'm going to end there. There's a few more slides about the regulation of immune responses and a particular cell type called a T regulatory cell, but that'll come up in some of the specific talks that we'll go through in the next few days. And so I'll return to these slides then. I'll end there, and I'm happy to take any questions.